So I'm Wes Spain, and I've got the pleasure of introducing our speaker today, Dr. Kathleen Vogel. Kathleen is an associate professor in the Department of Political Science and director of the Science, Technology, and Society program at North Carolina State University. Go Wolfpack. She holds a PhD in biological chemistry from Princeton University. She's been associate professor at Cornell University, served in U.S. Department of State's Office of Proliferation, Threat Reduction in the Bureau of Nonproliferation, and was a visiting scholar at Sandias Cooperative Monitoring Center and at the Center for Nonproliferation Studies at the Monterey Institute, which is now the Middlebury Institute, I understand. I was first introduced to Kathleen's work through an article she wrote a few years back for CIA's Studies in Intelligence. And that article led me to her book, which I'll be happy to share and promote for her, Phantom Menace or Looming Danger, in which she takes a real critical look at the challenges of accurately assessing the national security threats from bioweapons. As part of that investigation, she looks at the records of the Soviet Union, the Iraqi programs, and very interesting to me, she spends about half the book looking at the practice of national level intelligence analysis around bioweapons, primarily at the CIA, and points out those practices and processes and methods that sort of hamstrings more accurate assessments of the bioweapons threats. She concludes that study with recommending an alternative analytic approach that she argues would improve the production of knowledge in the bioweapons analyses. So I highly recommend the book to you. The section on the analysis at CIA is very insightful, and she obviously very well researched and very well sourced. It is spot on. And so today we're going to hear more about her work and her recommendations. She'll speak for about 45 minutes or so, and then we'll open it up to questions. Go easy on her. She's been traveling a lot with a delayed flight in Detroit last night, and she's off from here to Mozambique, so she might be a little tired. But please join me in welcoming Kathleen Vogel.
Oh, thanks. Thank you. Well, thanks so much. It's so nice to have this opportunity. I originally came out here to attend the American Political Science Association meeting that's happening in downtown San Francisco, and then had a chance to meet and talk with the rest, and sort of seeing that I was already going to be out here and would have loved to chat more and have this opportunity to come and share some of my work with you. So it's just great that it's managed to work out. And again, I really appreciate this full house, given that it is a holiday weekend. I'm sure you have many other things that you could be doing instead of sitting in this room. So definitely appreciate your time, and I definitely look forward to chatting with you this afternoon. So essentially, I'm just going to put this up. As many of you are aware, since September 11th and the anthrax attacks that happened shortly thereafter, there has been an increased attention in the U.S. government related to bioweapons threats. As I was talking to Brad Roberts a little earlier, a lot of this interest really started in the 1990s, where there was a flurry of activity occurring then, and that's kind of in some ways how I got into this line of work, is through part of that period of activity. And, but since September 11th, the anthrax attacks, there's been renewed attention. There's been a number of different types of programs and activities that have been instituted essentially to try and protect and guard against biosecurity threats. A lot of these, though, have been focused on probably, I would say, more practical and tangible matters, which I think are important. Things like securing pathogens, trying to increase disease surveillance efforts, developing more and better medical countermeasures. Again, all important areas. But I would argue, and my book talks about this in more detail, that there has been less attention and resources looking at how do we better try to understand the bioweapons threat, and how do we do a much better job, bring a much more holistic approach and more rigorous approach to trying to assess bioweapons threats. And sort of how these threats not only have technical dimensions, but how these threats are also shaped by important social dimensions, by different actors and different things that they can bring to their development of bioweapons capabilities. And this hasn't been an area of focus, I would argue. I think coupled to that, we also see that there's been kind of a long track record of U.S. government analysts, as well as folks outside the policy community, either underestimating or overestimating bioweapons threats. So I think there's a lot of work that still needs to be done in having a more refined way of studying these kinds of problems. And so essentially what my talk today is going to do is sort of outline maybe one approach that we can use. There could be many others, and I would welcome thoughts on that, but how we might be able to put together a more holistic way of assessing bioweapons threats. And again, with this aim at looking at how do we marry understandings of technical capabilities, technical issues related to biological weapons with then trying to incorporate then this more complex and harder to grasp kind of social dimensions that can shape the technical. So just to give a little background of kind of where I'm coming from a research perspective. So I'm drawing on this field of study called science and technology studies, which some of you may be aware of. Essentially, it's a whole field that essentially studies how science and technology is designed, developed, and used. And really looking at how these things are very much embedded and are influenced by the social or kind of environmental context in which science and technology take place. And so this area of research essentially does a lot of in-depth case studies. I would say that's probably one of the more strengths of this area, doing a lot of very micro-level case study analyses of science and technology in action. So looking at sort of what goes on in laboratories and really going in. Some folks in this field are anthropologists, so they would go into a laboratory as an anthropologist might go into the bush and sort of study native populations. And they go and they study scientific practice. They look and sort of see what are scientists doing in the lab, what kinds of tools and technologies, how are they using these materials, how are they talking, how are they communicating. But again, studying scientists and engineers as kind of a unique kind of social community and really documenting those day-to-day working level practices. Some of this work looks at sort of unpacking the more popular notions of science and technology as kind of straightforward and unproblematic, and really looking at sort of how science and technology work in practice and how often it's very messy to do experimental work, how often there's a lot of problem solving and trial and error and a lot of learning that has to go on as one works in the laboratory. And then other aspects of this literature look at not only kind of these micro-level laboratory practices, but then also look at what are the different kind of managerial organizational contexts that are required to make science and technology work. And then looking at, with these latter factors, looking at sort of what are the different factors, whether it's the large macro or the micro level factors, that enable some aspect of science or technology to succeed, or in other cases, looking at failure. Why is it that something, even if it was very technically, you would think on paper, which should technically work very straightforwardly, why is it that certain things never come to fruition? So again, these are some of the different areas that this literature explores, and really looking at trying to marry the social with the technical, and looking at both these micro level and macro level factors that can really shape outcomes. So I'm going to present to you today a few different case studies that I've looked at throughout the course of my research. I'm going to sort of just provide you a little bit of a snapshot of these in the sake of time, but in my book I have chapters devoted to each of these cases that go into a lot more detail. But I just want to talk about these cases because they're interesting to look at, again, to try and think about, you know, what is it that we have known about these cases, and then how, when you look at sort of these micro and macro level factors, how does that complicate the picture? Okay. So the first case I'd like to talk to you about is the Soviet bioweapons program. It was one of the cases actually that first really got me into this right after finishing graduate school as a postdoc. You know, the Nunn-Lugar programs on the nuclear side were you know, well underway at that point, and there was an emerging focus starting on the Soviets' bioweapons program. That's kind of how I got interested in this area. And you know, throughout the late 1990s, early 2000s, there were a lot of different people looking at the Soviet program, and a lot of revelations came out from defectors like Ken Elbach, talking about how the Soviets did have this very massive covert weapons program. It's probably considered to be the largest bioweapons program in history, and starting from the 1920s and continued on really until the collapse of the Soviet Union. And you know, the program was quite ambitious. There were, as you can see from this graphic here, a number of different institutes and facilities that were located across Russia and other of the Soviet republics that were involved in making different kinds of aspects of in the bioweapons program, and there were different kinds of formulations and new kinds of production methods that were developed, and they even did try to apply different kinds of genetic engineering techniques and technologies to try and modify pathogens. So there's a lot of different work that was done in this program. And a lot of times, I would say in the 1990s and 2000s, a lot of the focus was on just the worrisome of this threat, that it was so large and they had done so much over this time period. But one of the things that you find, though, if you look much more closely at sort of what actually went on in some of these laboratories, that the program was actually quite fragile and it was uneven in its technical progress. There were a lot of different challenges that the program faced, and so the end result actually was less than what you might presume from just looking at kind of numbers, from personnel to materials to facilities. It's a much more complicated picture that actually emerges later. And so let me talk to you about one facility that I've looked at in some detail. This is the Stephanyshev Facility, one of the main mobilization plants that the Soviet Union established in Kazakhstan. And this was a facility that was set up in the 1990s, essentially as kind of a covert production facility, essentially to try and prevent us from knowing what the Soviets were doing in this area. They decided to establish this new capability in Kazakhstan. And this facility was given the task essentially to create a new anthrax weapon. And this new weapon would be based on the Soviets' existing anthrax biological weapon. And to do this, when this facility was starting, they received a lot of very detailed classified documentation from the Soviet Ministry of Defense about the existing anthrax biological weapon, on protocols and other kinds of production methods to make this weapon. And what's interesting is that although they had this very detailed technical information, they had a lot of problems with actually even just replicating the existing methodology at this new facility, let alone try and modify it. And so what ended up happening is that because of all these technical challenges, a decision was made essentially that they needed to basically bring to this facility basically the top anthrax experts of the Soviets' bioweapons program across all aspects of the production process. So the anthrax researchers, the bench scientists, along with the Soviet production specialists to the weaponization specialists. And these were all key experts that were brought in from Russia to this facility, essentially to try and make these protocols work. Because they had sort of that resident knowledge of having worked on this older method previously. And so what ended up happening is that when they brought these individuals over, it wasn't a sort of a quick and dirty, they were able to get everything running. There was still quite a bit of adaptation that had to occur to essentially adapt these protocols to work in this new facility. This facility was quite different from the other facilities where the existing protocols were made. Some of the equipment was different, how it was configured was differently. They had different types of equipment that they were using. And so all of these things, they had to adapt all the protocols to work. So it was not only just bringing them over and then quickly getting it up to speed. There was a lot of trial and error of how to make all this work in this new facility. They also, the manager of this facility also created some new kind of interdisciplinary work groups at this facility to try and promote learning and integration of knowledge at this facility. And so there was still 3 more years of work that they had to devote just to try and get these existing protocols to now work and then to modify them to work at this new facility. So overall, what's interesting is although this was a technology that was based on a really existing, proven kind of method, it still took them longer, essentially, than the Soviets' first nuclear weapon, which is kind of interesting to think about. You'd think it would actually be quite easier to do, but actually took quite longer. And so essentially, the take-home message, from looking at this case study is that it wasn't just sort of the information, the fact that they had these protocols. They needed all kinds of different new kinds of knowledge from how you adapt these protocols to work in a new setting. That required new kinds of know-how, hands-on knowledge, skill sets to do that. Different kinds of management arrangements at this new facility, different kinds of training for employees that were working at this facility, et cetera, to actually then ultimately to create this new Soviet anthrax weapon, which they were able to do, so that was possible. But there was a whole set of circumstances and factors that was needed to allow that to happen. So another aspect of this that I wanted to sort of talk to you about is some work that I've done with Sonia Ben-Agram-Gormley at George Mason University, where we've continued to look not only at Stephanyshev, but some of the other Soviet bioweapons facilities. Again, to look at, what are a variety of different factors that can shape bioweapons development at particular facilities. And so what we did is we looked at a couple of the key research facilities in the Soviet bioweapons program, the Obelensk facility, as well as the Vector facility. And what you see are some very different kind of institutional organizational factors that actually then ultimately shape how successful each of these facilities was in their bioweapons efforts. So, for example, on the Obelensk side, this was very much a military research institute. It was led by a general. What's interesting is at this facility, there were a lot of tensions between the more civilian scientists that were working there and the military staff that were based there. The director, General Rakov, who led this facility, was very much a micromanager, and he wanted things to follow a very strict kind of hierarchy. He was very much micromanaging various levels of the research that went on at this facility. And so there are a lot of tensions with him essentially exerting this control down to the bench level at various points. At this facility, a lot of the research was very compartmentalized. Scientists working in adjacent labs often didn't know what the others were doing. This was a particular secrecy practice that was instituted at this facility, again, largely by this director. The scientists had very little scientific freedom. It was very much sort of a task that they were to accomplish, and that was what was to dictate their time. They weren't allowed a lot of creative freedom to explore different avenues for research. And this facility also was very much centered on trying to maintain essentially classical methods. They really weren't interested in innovating. And the director really resisted, for example, trying to introduce, new genetic engineering techniques. He really was not interested in that. He just wanted to keep to the task and focus on essentially what they had always been doing. If you contrast that with the Vector facility in Novosibirsk in Siberia, you see a very different kind of model for how bioweapons research was conducted. This director at the time was Lev Sendakchiev. He came from essentially the academic community, kind of more the civilian side. He was very much a supporter of having a lot of academic freedom in his facility. And he very much encouraged his staff essentially to explore innovative new areas. And he gave them rewards and different kinds of perks for essentially innovation. He sponsored a lot of different kinds of teamwork, encouraged people to share information across the labs. And so what you see here at this facility is you see a lot more innovation in terms of what they were actually able to do compared to the Obelensk facility. So again, it's kind of interesting to think about sort of how these different kinds of managerial styles, organizational styles, can fundamentally shape the type of work that actually comes out of particular facilities. And what you see is the Obelensk facility, in terms of their output, they definitely were hindered in their sort of bioweapons related work, whereas the Vector facility had a lot of different kind of innovative approaches that came from that facility. Kind of scroll through some of these. And again, so it's interesting to look, if you kind of take this macro level picture, kind of moving from the institutional organizational approach, if you kind of look at a big kind of macro level picture of the Soviet bioweapons program, you see that they had a lot of people, a lot of institutes, they had a lot of resources. It was very much a high political priority to have this program. But you also had these really interesting phenomenon that you also see occurring across the bioweapons program. So for example, one of the macro level problems that you see occurring across this program is there was a lot of political intervention into the work. You had, for example, a lot of technical decisions or decisions made about technical personnel, for example, being made by Communist Party officials. And sometimes individuals will be promoted due to their loyalty to the Communist Party and not necessarily because of their technical expertise or their technical skills. There's also a lot of pressure to meet kind of the Soviet 5-year plan, even when, let's say, how the plan was laid out on paper for some of these facilities didn't make sense. But there was this pressure to do the plan, and then what that ended up happening, and I don't remember if I have it here, is essentially then some of the scientists in these fields, what they started doing is they started creating false technical reports, essentially to show on paper that they're meeting the plan, but in reality their experiments weren't working at all. And this was facilitated by the fact there was a lot of secrecy and compartmentalization across the program, so people didn't always know certain details about experimental, facts. There are other aspects of the fragmentation in that across the program there really wasn't a good integration system that was going on, so that folks from the research side to the production side to the weaponization side, there wasn't really good connectors often. In some cases there were, but there were disconnects kind of across the program. You also had some economic issues. Again, the Soviet supply system, for example, often there were a lot of problems in getting access to necessary materials for experiments. And so this caused a lot of problems in terms of making progress in some facilities. And then there are also disconnects, with the military. There really wasn't, this is something that comes out from actually Milton Leitensberg's really extensive book that was published on the Soviet bioweapons program, where there really was no military doctrine for how these weapons might have been used over the whole time span of this program. And there really was no strategic delivery capability for this. And again, it's hard to imagine, given that there was such extensive resources and this was a high priority. All of these factors are peculiar. But again, it shows, I think what this shows is there are a lot of different kind of micro-level factors and forces that could come to bear on a weapons program that one needs to be thinking about and incorporating into assessments of capability and progress. Okay, so the second case that we can look at also is a state-level program. This is Iraq's bioweapons program, and as many of you know, a lot of revelations about Iraq's bioweapons program essentially came out, after the first Gulf War. Revelations that Iraq did have a bioweapons capability, even though they had long denied it. And you hear pictures of, one of the main production plants, the Al-Hakam facility, that UN weapons inspectors eventually destroyed. And then, a lot of concerns about where this program is going. There were a lot of concerns building throughout the 1990s and early 2000s that Iraq was continuing to hide its bioweapons capability. And then sort of in the late 1990s and early 2000s, you see this focus on, were they evading this through creating some kind of mobile bioweapons capability? Were they somehow able to move, going from like a fixed production facility to essentially being able to move this facility around, let's say on railroad cars or on trucks or other things? So this was something that was people were wondering about, because there was not necessarily evidence of a substantial bioweapons program, but always suspicions that there was something going on. And so again, after the 2003 war and the Iraq Survey Group and others who have done, research on sort of the status of Iraq's bioweapons program, it's interesting to see what comes to light. After the war, what we see, and this definitely comes out through the Iraq Survey Group report, is that Iraq really didn't have an offensive bioweapons program essentially since the mid-1990s. And that was really due to the fact of these intrusive weapons inspections, the sanctions, which had really made it problematic to maintain a program, and then a variety of different aspects of corruption where Saddam Hussein was essentially putting more money into paying off his security forces than actually investing in a weapons program. What you see also, after, again, through evidence that comes out after the war, that Iraq had a lot of technical problems for bioweapons development, as Sonia Gormley's book essentially talks about this, where, again, the Iraq program had a lot of kind of cyclical, disconnected efforts kind of over a multi-year period. Similar to the Soviet program, it was very compartmentalized. Teams and scientists didn't necessarily work together or work in an integrated fashion. And then you had also this macro level thing of political intervention and fear that kind of characterized different aspects of the program. Similar to kind of the Soviet program in some cases, where kind of the leader wanted certain kinds of results, and led to then all these kind of bizarre practices where scientists would then actually do false reporting or create false technical reports to try and prove that they had accomplished things, but in actuality, very little or nothing had been accomplished. One of the things I also talk about in my book related to the Iraq, what we know now versus what we know then, is some of the problems that the intelligence community had in trying to assess Iraq's bioweapons program. And some of the things that I talk about, again, through various chapters in my book, is the problems that intelligence analysts had in not scrutinizing human intelligence. as well as the fact that the CIA analysts who were involved in these assessments really didn't take into account kind of these broader, the role of sanctions, the role of the weapons inspections, the corruption aspects. That really was not at all factored into essentially how they were assessing how those things might have an impact on the weapons program, for example. And one of the quotes that I found as I was doing some of my research was this report put out by Richard Kerr, who essentially was talking about what he saw as a fundamental problem. And he was brought in by the CIA to essentially do an independent outside assessment of what were some of the intelligence problems leading up to 2003 war. And essentially what he talked about, which is something that I've been interested in, so when I saw this and what I was seeing in some of my research, I found it resonated really well. As he commented, there was very little sort of integration of folks who are essentially doing the technical assessments versus others who maybe might have understood the Iraqi context. There was just very little integration of that kind of knowledge together. And he felt that that was a major problem in, again, creating these more abstracted, kind of technologically advanced ideas about what Iraq was doing in its bioweapons program, which really didn't connect with what was actually happening on the ground at all. Okay, so let's then now move from those two kind of state programs to now talk about maybe a different case, looking at concerns about advances in biotechnology, advances in the life sciences. And one area that I've looked at is this whole area of synthetic genomics that has gotten a lot of concern over the past, gosh, now it's been now 10 years or so when some of these concerns first started emerging, and you see it now in different aspects of the whole synthetic biology debate and discussion. But this is one case that I got very interested in 2002. It was a paper that was published in Science by Eckhard Wimmer and his researchers at the State University of New York at Stony Brook. And when they published this experiment, this was an experiment that was one of the first, well, actually it was the first synthesis of poliovirus through chemical means, artificially. And it was published in Science and got a lot of attention, including being picked up by the New York Times at the time. And there was a front page news story about this paper. And it just so happened that on that day when it came out in the Times, there was a congressman who actually read the Times and basically became really concerned about this paper being published. And again, you can think about the time period, 2002, right after the, not too long after the anthrax attacks. And he became very concerned that essentially a terrorist might be able to download this paper and essentially maybe recreate this experiment, release poliovirus, or maybe even manipulate it to create some other more deadly form of virus that could then be released in a bioterrorist attack. So then this generated, when this congressman raised these concerns, this generated all kinds of policy attention in Washington. The White House started looking at this. There were a lot of discussions with this paper, and you see this actually continually in the bio realm. This continued question about should we be publishing the materials methods sections of this kind of work? Or is there a need potentially to redact, to take out some of the experimental details to prevent someone from replicating this kind of work? And this concern first emerged with this experiment, but again, you kind of see it, and it's continuing to, depending on whether we're talking about the H5N1 or some other kinds of experiments, every few years, this issue of should we be publishing these papers? Should we be publishing them as we usually do in science? It's an issue that in the biological sciences, you see it's a continuing tension, and it's not easily resolved. And I think we're going to see more of it as we go forward. But let me tell you a little bit about this experiment. I talk about it more, again, a whole chapter in my book where I talk about this experiment. So here's essentially a schematic of this experiment. And I'm not really going to go into my book. I talk about different aspects of this work. The main concern, I would say, that generated all the different policy media attention of this experiment essentially focused on the top part of this experiment, which focused on, for example, that the information about the viral genome sequence was available, that you could get it off of the internet, the fact that you could use, for example, a basic computer to convert this information to a more usable form, to then allow you to then commercially buy short pieces of DNA that you could then stitch together using some pretty common molecular biology techniques to then eventually make this virus. So again, a lot of the media and policy attention focused on on the information and the materials and some of these simple molecular biology techniques to kind of stitch these pieces together. What you didn't see, however, was a lot of focus on this bottom part of this experiment. And that's as I started looking into this experiment more in detail, what I found is actually this bottom part of the experiment is actually quite crucial to getting this experiment to work as it's published. And what I found is, again, in looking at, and this is, again, this science and technology studies approach of you study experiments in practice. So what I did is I went in and I talked to this research group, the Wimmer group, did a lot of detailed interviews and did some observations in the laboratory as they were kind of doing some of this experimental work to look at sort of what was required to actually do this experiment as published. And what I learned is actually there's quite a bit of specialized know-how to make these HeLa cell extracts, which is what then eventually catalyzes the formation of the virus at the end of the experiment. This was something which was not evident when you read the entire science paper, even if you look up all of the references, which I did, and read through all the detailed references in terms of the protocol and what's required. No one really talks about how this is kind of a really crucial and in many ways a very tricky part of getting some of this experimental, this experiment to work. And so this kind of came out in interviews as I was sort of talking about, well, tell me about kind of this practice and what you do. And so let me show you in the next slide one of the interview quotes that I had with one of the researchers involved. And to make these HeLa cell extracts, this is what one of the researchers said. She says, making it's very irreproducible. You can take these HeLa cells and make them today and they'll work fine. And you can make the HeLa cells tomorrow and they talk about how, there's a lot of variability. And she says, we don't really know what's in the extracts. Translations always work with these extracts, but you don't always get virus. And she talks about, we've tried a number of different things to try and figure it out. It has something to do with cells. She says it's maybe not something with replication actually, but it's the encapsulation, the formation of the virus. Maybe it requires something that's possibly lost during making the extract. Who knows? It's just a guess. It's not only a problem for us, it's a problem for anybody who works with the system. And she says, if you've got one extract that doesn't work, it will never work. And when she mentioned this was very surprising to me because I'd never heard anyone talk about this aspect and the difficulties of this part of the experiment. And I sort of went on to kind of ask a bit more, kind of probe that a little bit more. And it's not that making these extracts is something that was new and foreign. This is a technique that this research group had actually pioneered for over 15 years by that point. So it's not kind of some new novel technique. There's just something that's really particular to this aspect of the work. One of the things that, as we talk sort of more about, well, what are some of the different things that come into play with this particular technique? She talked about how, in terms of making these HeLa cell extracts, they use this thing called a glass Downs homogenizer, and you could think of it as a fancy mortar and pestle, where essentially you put these HeLa cells in there, and what you're trying to do is crush them to release some of this cytoplasmic material that then you use for to make the viral part of the experiment. And so she was talking about how, this sometimes can be a very tricky kind of tool to use. And one of the things I was interested in probing is this something that's maybe unique to this group? Are other people who work in this area, other scientists in the field, are they also encountering these difficulties? So then I interviewed different, essentially competitors of this research group to sort of hear, are they facing these same kinds of issues? And so I conducted one interview with Bert Flanagan at the University of Florida. And sort of he talks about, as you kind of read through this quote, essentially he talks about there's a particular kind of art with using this tool. That if you don't sort of do the right tension in terms of essentially trying to crush the cells, you'll either break the device potentially, or essentially you'll degrade the cells in the process. So he was talking about there's very much kind of this technique or this art form with actually using this particular tool. He said that's something that's, you can't really read from a paper. It's something that you actually learn while you're in this community of scientists who actually use this technique, that you learn from experienced people using this technique. And one of the things that he and other polyvirologists that I talked about who do this kind of specialized work is that, again, because there's a lot of variability, they try and control it in different ways. And there are all these kind of interesting practices that they've instituted in their labs to try and control this variability. And so, for example, they've talked a lot about trying to keep certain things the same in the lab. And it's these really interesting range of things. So, from a sameness in materials. So I remember for part of this experiment, they use a bovine fetal serum. And essentially, one of the labs says what they do is they essentially test different lots of the serum from different companies. They find one that actually are able to make virus from this experiment. And then what they do is they buy 2-year quantities of the same lot, essentially try and keep that variability throughout this kind of time course, because they've found that if they've bought different lots and things, the experiment fails. So they try and sort of keep some of these buying practices the same. They also talk about, in terms of sameness, the importance of keeping the same people doing this kind of work. So the person who's actually not listed on the paper, but one of the key people involved in this experiment was a lab technician who'd been in the lab for 15 years, and she's actually the one who made the helicell extract. And this is something that she's been doing for many, many years. And again, she was actually central to getting this experiment to work, but she's not listed on the paper. So unless you went in and really looked to see what is actually happening in the laboratory, who's actually doing some of the work, you wouldn't really know that that person was quite critical. And similarly, in terms of other practices, if they're grad students doing this work, they have to be really working with the system continually. It's not something that a grad student can just do as a one-off kind of experiment. And then requiring a lot of disciplines. They were talking about how you basically have to be very anal retentive to do this kind of work. It's not a general virologist, someone who works on DNA. They sort of talk about how it requires a very particular kind of focus and set of disciplines across the whole experimental components that really you have to pay attention to that. So one question that often I think comes up when I talk about this is, so that's one experiment. We can think about, that was 2002. Are there new kind of innovations or new things that might actually have changed how difficult that might be? And again, we can look at other kinds of synthetic approaches that have been done since that experiment was published. And what's interesting, that you see, across these, and when you're talking about synthetic genomics, for example, is you see kind of a larger set of resources and kind of people that are required to make these kinds of experiments work. You even see specialized whole gene synthesis companies being brought to bear on these kind of newer approaches. And if you read different accounts of some of this work, you see they still have to do a lot of different kinds of problem solving. There's a lot of specialized know-how, a lot of different kinds of resources and time that's required to get these experiments to work. And that's not to say that certain kinds of standardization or certain things may become easier in some aspects of these experiments, but it still requires some work. And again, what's fascinating to think about some of these different synthetic approaches is that some things are able to be standardized and some things are not. So for example, for the Wimmer experiment, that particular technique, even today, and talking to folks recently, if you want to do that particular experiment, the march of time, new technologies is not, if you want to do it as it's published, that hasn't helped you. It's still going to be difficult to try and replicate that experiment. So with that, I would say it really depends. You have to look very closely, like, are there particular, very specialized kinds of skill sets that are required to make a particular experiment work? In the polio experiment, there is a very unique set of experiments. That doesn't mean, or set of skills. That doesn't mean that every experiment is going to be like that, but you have to look at it very closely to be able to tease that out. So again, for this, for something like the synthetic genomics kind of work, I would argue, there's been a lot of focus on how potentially soon anyone can do this kind of work. Again, what you see if you look at some of the different approaches, it's getting much more difficult and complicated. And I would, again, argue that this would then lead you to maybe think about, what would be required to sustain that kind of effort. And also to look at, these past examples which sort of talk about, if you're going to do this more complicated, team-oriented, more complex kind of science, what are the different kind of social factors involved? What kind of training, what kind of organizational, managerial practices do you have to bring to bear to make that work? And there have been different kinds of literature. There's, my field, science technology studies. There's also a whole set of different references in sort of the team science literature and communications that have looked at interdisciplinary science teams and different pharmaceuticals, biotech, that sort of write about what does it take to get these interdisciplinary teams to actually work successfully. And I think these literatures can be really useful too as informing this kind of assessment. So this is what I just pulled from my book. And essentially, kind of from all this, essentially in my book, what I try and argue is that we need a different kind of framework for assessing bioweapons threats, where we look at these different socio-technical dimensions, where we look at these kind of hands-on skill sets and different kinds of know-how practices that are actually required to make certain kinds of experiments or technical projects work. And how oftentimes these involve not just a sole individual, but oftentimes communities of people, teams of people who actually are involved in making an experiment or a technology work, and how there are different kinds of organizational and managerial factors that have to be brought to play. And with that, essentially what I argue is that we can't just assume that every advance in science and technology is going to directly lead to an increased threat. It varies, and I think you have to really try and tease out what are the complex combination of factors that might increase the threat, and in other areas that actually might not change the threat at all. And there are different threat trajectories that can be possible, and to really think about that. So with that, I'll just kind of end and, essentially sort of, hopefully illustrate that, when we talk about, biological weapons threats, we have to sort of think about how, when we talk about any aspect of science and technology, it's often kind of messy. There's a lot of contingency, there's a lot of different kinds of social dimensions that can shape the end result of science. And because of this, I think we need to devote more attention to trying to tease out these different social dimensions and how those then would connect to enabling a particular kind of capability, enabling a particular actor or set of actors to essentially accomplish a particular kind of threat. I think we also need to learn, we need many more case studies of success and failure in this area, not just in terms of threats, but in looking at sort of S&T in general. So looking at, for example, in detail, Ventor approaches and really doing more detailed analytic studies of what does it require for Ventor to create a synthetic genome, and really look at that in terms of the day-to-day practices, the time scales of what's required. I think there are a lot of assumptions that people make about that, but there's very little work that actually do the heavy lifting of interviewing, being in the lab, watching what goes on, and really making a more empirical assessment of that. And then with that, to then, when you have enough empirical cases, then to try and then make some sense out of the historical cases and how that connects to the contemporary. And then finally in my book, I talk about, to do this kind of work, you do need interdisciplinary groups of people. You need people who understand the science and the very detailed aspects of technical practice, working then along with people who understand S&T managerial structures, what is important for S&T innovation from an organizational perspective, and really getting both of those sets of people to be in conversation with each other. And I think if you're able to do that, then I think that'll give you a much more robust set of cases then, that you can start creating a more refined spectrum of what the threat actually is. Instead of it being the sky's falling or there's no threat, that you probably see more of there's a spectrum. Certain things are probably more problematic than others, and it would be useful to try and parse that out through different kinds of case studies. So with that, I will end, but I definitely would welcome any questions that you might have.
Thanks.